Okay, hello everyone, and welcome back to sugars and nucleic acids. This is part 2A, and I've put a picture of a Halloween, pumpkin from Halloween because, well, it's kind of a cool image I found online, but it's also, as you can see here, it looks like a double helix. So we're going to be talking about DNA a lot in this unit. This first lecture will be probably a little shorter than usual. We're going to talk about the prebiotic synthesis of nucleosides and some of the properties of oligonucleotides. So here's the outlines divided up into 3 parts. Again, that's our first one, and then after that we'll talk about biological enzymatic synthesis of oligonucleotides, followed by the chemical synthesis of oligonucleotides. Okay. So last time we talked about a lot of chemistry of sugars, and what I want to focus on is how nucleosides could have perhaps been made in a prebiotic world, how they could have been coupled to sugars. So you've seen already that heterocycles could be made, things like purines, adenine, perhaps you've looked at guanine, thymine, uracil, some of those heterocycles, but how did they actually couple to sugars? Okay, so we have seen how the intermediacy of the resonance stabilized oxonium ion accounts for fast cell substitution at the anomeric center of a sugar. So, prebiotically, it's quite conceivable that you would have some type of sugar, and then attached here you would have some type of leaving group. So this is OPP. It doesn't stand for the Ontario Provincial Police, in case you were wondering. It stands for this, this diphosphate. And this phosphate, as you know, is actually a good leaving group because you can actually move electrons through the double bonds. So that's actually a fairly decent leaving group. So it's actually quite reasonable to form this oxonium or oxocarbenium ion. And I guess one of the questions is, well, how were nucleosides formed, right? So you can imagine that you have a good electrophile. Was this formed from ammonia being one of the precursors that was around during the prebiotic era? Could this have acted as a nucleophile? Yeah, perhaps. Another thing that could have been made perhaps are these types of heterocycles like quinolates, right? These were very similar to some of the structures that we saw like NADH, that could have been formed during like a Desaulniers type reaction. So these types of heterocycles, these pyridine-like moieties, could have also participated in this type of interaction. So you can imagine these coupling to sugars, and then this could then be reduced to something like NADH. And furthermore, the NH2 could act and serve as a nucleophile to start the synthesis of nucleosides. So could this process have occurred in the prebiotic world? Well, it's definitely, it's definitely possible, right? So some researchers have set up some experiments to try to see what could have happened prebiotically in terms of the generation of nucleosides. So if sugars are formed, and we've learned that these can be formed, through the elongation of cyano groups to aldehydes, it's possible that perhaps in hydrothermal vents or in the presence of minerals acting as a Lewis acid, you could actually have this group coordinate, if you don't have a phosphate or a pyrophosphate, leading to an oxonium ion, and then you could get some of the heterocycles coupling to that oxonium ion to generate nucleosides. So perhaps the nucleosides, or perhaps the nitrogenous bases, were made earlier and then they coupled. So minerals like apatite could perhaps facilitate this reaction. Phosphates, which are good leaving groups, they can be activated by catalysis. You now have a good leaving group, and perhaps this could have also led to the synthesis of nucleosides. So no one really knows for sure what exactly happened. However, one thing that we do know for sure is that nucleosides are actually very stable compounds. And this should come to no surprise. People have been examining DNA in forensic cases where DNA has been around for years, dozens of years. And if you think back, or if you think even further to, dinosaurs, we can access the DNA there. So these structures are inherently very stable, and that's a really good thing. So nucleosides are quite stable. And if we think of why did nature pick a nitrogen to react here when you could think of oxygen or maybe halogens going, there's actually a very weak or much weaker anomeric effect with nitrogen compared to oxygen. Okay, so we learned about the anomeric effect that stabilizes, it stabilizes that interaction, right, through the donation from the oxygen into the sigma star CO sigma star bond. However, nitrogen is less electronegative than oxygen, so it's less willing to accept those electrons. So that can actually make it more stable. So it's not going to pull as much and not be as good as a leaving group. Furthermore, we've learned about aromaticity in some of these heterocycles. So it turns out that if you actually try to protonate something like thymidine or some of the other nucleosides, you actually lose aromaticity. So it's actually hard to protonate. These electrons here actually participate in the cyclic aromaticity, and therefore protonating it would make it very unstable. So it's very difficult to protonate. So that's another reason why they're so stable. Here is another sort of illustration showing what would happen if you were to form an oxonium ion. So if this were to happen, you get an unfavorable charge separation. Remember, this oxonium ion or oxocarbenium ion, those mean the same thing. This oxygen has a positive charge, right? That's not very favorable, pretty reactive. Furthermore, the nitrogen here has a negative charge, right? It would rather be the other way around. If you think back to the electronegativity, if you compare the electronegativity between carbon, nitrogen, oxygen, fluorine, so this is the upper right hand of the periodic table, as you go from left to right, you increase your electronegativity. Okay, so oxygen is more electronegative than nitrogen. So this is unfavorable charge separation. So the anomeric effect, something like chlorine, oxygen, nitrogen, remember that the glycosyl chloride prefers Cl axial. And in all these cases, the nucleosides are actually in a beta orientation. Secondly, as I just alluded to on the previous slide, it's actually very difficult to protonate because the lone pair of the nitrogen is part of the aromatic sextet. So in fact, if you do protonate it to try to make this now a good leaving group, you actually destroy the aromaticity, and that is unfavorable. And so it's actually, it's not impossible to do this, but you need some very, fairly harsh conditions to do this over an extended period of time and to any significant extent. So that's just a little bit of information on these different types of molecules, of how they could have been made prebiotically. And what I'd like to do is just switch gear a little bit, because what we're going to be focusing on for the next couple sections is how to make oligonucleotides. But before we study oligonucleotides, that is the polymer of DNA and RNA, why would we want to bother studying them? What is so interesting about them? Okay, so one thing that perhaps some of you have done before in lab is something called a polymerase chain reaction. Okay, polymerase chain reaction is a technique used to amplify DNA. It's also used to quantify low levels of DNA or RNA. So it's a very versatile tool, and it's even more important today as we try to find new ways and new tools to test for COVID-19. COVID-19 testing is all done by PCR. Okay, so it's a very robust, very accurate method. In order to do PCR, you need to have access to primers, short oligonucleotides that can be specific for your particular gene. So it's useful for PCR. It's also useful for molecular cloning. So if you're trying to express proteins recombinantly, you need a plasmid vector in order to generate your desired protein of interest. You're going to need oligonucleotides for that. So things like DNA recombinant technology. Some other interesting things are things like gene silencing oligonucleotides. Okay, and I'll show you a couple examples on the next slide. Okay, so these are oligonucleotides that can directly bind to or interfere with the messenger RNA and either prevent the protein from being made in the first place. Okay, and there's some approved oligonucleotides that are therapeutics. And these are relatively short oligonucleotides, roughly 20 nucleotides in length. Gene therapy, you may have heard of a technology called CRISPR-Cas9. I won't be talking too much about that in this class. I'll talk about it more in my next class, next semester. But this is a gene editing technique that was developed or discovered in 2013. And for that, you need to generate oligonucleotides. Oligonucleotides are also a platform for chemistry, being able to use catalytic oligonucleotides to perform reactions. So in protein worlds, we have enzymes. We can actually engineer and we can design molecules that are oligonucleotides but can catalyze reactions. So we have molecules called RNazymes, DNazymes, and very similar to proteins, these assemble in a unique three-dimensional structure and they can catalyze very specific reactions. It's important to do structural studies in order for us to understand the structure of oligonucleotides, especially if they're are acting as potential next generation therapeutics. Oligonucleotides can have very interesting structures, so being able to synthesize large quantities and study that using various biophysical tools is very, very important and very useful. So this list can go on and on and on, but there's a great need to study and to be able to generate large quantity of oligonucleotides. This is actually a very interesting and very exciting time to actually be in the field of oligonucleotides, because there actually is a fairly large resurgence and confidence in oligonucleotide therapeutics. I'll just highlight a few here. You know, in the late 90s, there was an antisense oligonucleotide called Vitravine that targets a specific virus in the eye. That was FDA approved. There's a few other oligonucleotides. One is an aptamer that targets a specific protein called VEGF. But you can see that after 2004, there really wasn't a lot in the oligonucleotide therapeutic landscape. And it wasn't until 2016 when Spinraza came on the market, and this treats a very rare disease. And then shortly after, 2018, there was siRNAs, and this was a short interfering RNA, which was the first FDA-approved RNA-based therapeutic. And this one's called patisiran. And then there actually has been another siRNA that was FDA approved in 2019, just last year. And there's also a collection of other antisense oligonucleotides that have hit the market as well, in addition to this one that I forgot to mention, but that was developed in 2013. So there was, like I said, a little bit of a gap, about a 10-year gap. And the oligonucleotides, there's been efforts to try to use siRNAs to combat the coronavirus, for example, gene silencing oligos, perhaps taken as a respiratory inhaler to target the coronavirus. So you could think of something like this. There's some very interesting developments right now, and this is done by a company called Mrna in the United States, developing vaccines. And it's an mRNA vaccine where a patient would potentially take this mRNA, and then the protein would be expressed in the body, and then you could elicit an immune response to hopefully offer some protection against something like the coronavirus. So the idea is that you could have an mRNA that codes for a portion of the protein that's involved in triggering an immune response, so you could build immunity if entire virus comes and attacks. So these are just some examples of therapeutics. We'll touch more upon these in a couple weeks, and certainly in the second part of the course. And before we end, I just want to talk to you about just some of the some of the definitions, and just so everyone's clear in terms of what ribonucleosides are and what deoxy ribonucleosides are. So ribonucleosides are molecules that are ribose, and I'll just draw an example here. It's a ribose sugar that's connected to a heterocyclic base. What I'll do is I'll draw uridine as an example. So these four bases are some of the main players in RNA, messenger RNA. They are certainly not the only ones. RNA is actually quite versatile, and you can think of like transfer RNAs, you can think of ribosomal RNAs. There's a lot of other types of RNAs like microRNAs. RNAs can come in all different types, shapes, and forms, and there are actually over 100 different types of chemically modified RNA nucleosides. So it's kind of interesting, why would nature decide to go beyond these four main bases for different applications? So the ribosome, for example, has dozens of different modified ribonucleosides. I really don't have too much time to talk about many of these different modifications, but I will just give you one example of a modification here. Since I just introduced uridine, this here is actually an isomer of uridine called Pseudo-Uridine, and it's present in some areas of the ribosome. But what's interesting about Pseudo-Uridine is that it is formed through a carbon-carbon linkage here, and essentially now you have two amino groups here. So in some ways this group has come off, and then it's reconnected at this carbon here. So this gives Pseudo-Uridine some different flexibility, different abilities to hydrogen bond, and offers structural advantages. And this carbon-carbon nucleoside also gives it even more inherent strength compared to a carbon-nitrogen glycosidic bond. DNA, in contrast, these are deoxy ribonucleotides. So I'll just show you what that looks like. So you don't have a hydroxyl group at this position. And I'll give you an example of thymidine, which is similar to uridine, except that there's a methyl group here. Okay, so there is no like deoxy uridine. What we have is deoxy thymidine. Okay. And lastly, what I have here are some of the structures of these ribonucleosides. So again, we've seen a lot of these already. I wouldn't expect you to memorize these, but I think you will become quite familiar with these. Cytidine, uridine, these have your two pyrimidine heterocyclic bases, and the adenosine and guanosine have your purine nucleosides, or your purine heterocyclic bases. And then the deoxy ribonucleosides, as I mentioned, just have, the two protons here. Okay, so they have just the presence of this hydroxyl group, as we'll learn with RNA, can have significant stability and structural implications when we're dealing with an entire oligonucleotide. Okay, but for now, that's it, and we'll see you for part 2B soon.